Okay, so today I'm going to explain some brief idea about the bioinformatics because it is the new field for some people and also some big data, how we can use these bioinformatics and big data to um, help to for this COVID-19 pandemic situation. So actually, what is bioinformatics? Bioinformatics actually combined field. It is a multidisciplinary field of science, and it's combined of engineering, biology, statistics, medicine, computer science. And the bioinformatics is really important because they use like computational knowledge and collect data from different disease related, also big data, and then it is we like the bioinformatics people interpret those data using different technology, develop software, develop method, model, algorithm to interpret those data, and also to predict or identify some drug that could be useful from the existing drug for the new disease. Now we are waiting for the vaccine of COVID-19 or the any drug that could be useful for COVID-19 patient. But how we can get those drug or vaccine? Who will develop this drug and who can give the idea or information to generate this drug? This bioinformatics is really important to develop this drug because to develop the drug, we need in details of different things from virus to human. And we need a lot of information, interpretation, which is not easy task or not a small, tiny task. So how we can, like the bioinformatics can help in this case. So the virus is infected human. And so for this reason, we need to know the virus mechanism for development of the vaccine or drug or to prevent this one. Also, we need to know the infection mechanism, how it is infected the human, in which organ, how much affecting in which organ, and also the effect after the infections. And also, we need to know the process of drug development considering all the information of virus and the human body. So it is not an easy task. It's not a tiny task. And it's not a small data. It's a very big data, big information. And now it is possible to develop the drug, but we have a lot of information, but the information is changing continuously. That is the problem. So for example, I'm starting to talk on about the virus, the SARS virus. How do we know, or how do we know this is a coronavirus? So we know this one from the DNA or the whole DNA sequencing of this virus. Although it is very small, 26 to 32 kilobase in length. 32 kilobase means 32,000 bits. Bits means if we see here, the each bit means on letter here. So it's a computation. When you say the bits, it's like a computational things. So to understand the virus, okay, it is a coronavirus. Why it is not the old coronavirus? So after sequencing this one, we get the sequence of what? The sequence of this letter, like a letter. And from this letter, we need to understand it is a coronavirus or any other virus. So based on this sequencing, we are now sure that it is a new coronavirus because this sequence is similar to the old coronavirus COVID-1, but some difference than the old coronavirus. Also, we like the people compared with the old like other virus or any other parasite or anything, and then found that no, it is different than those, but mostly similar to the old coronavirus and little bit similar to the MERS. But it is not a small task. It's like the general people, we cannot do manually this one. We need some sophisticated tool. We need to develop some algorithm. We need to use those algorithm, even machine learning or deep learning model to compare this sequence to identify the similarity, dissimilarity, and to identify what is the difference with the previous virus. So the bioinformatics, like people who is like the many researchers, they developed some algorithm model to identify model and tools to identify these difference and alignment approach. On the other hand, we know the evolution. So, for example, we know there was another pandemic which like its name was Spanish flu. But actually, it was not generated from originated from the Spain. It was originated from the China. But how do we know about it was originated from China? From this virus sequence, for example, here if we make the sequence of the virus in different time points, and if we compare each other, we can find out which virus came at first, because the virus always changes its structure. Like coronavirus, it is happening. It is like changing its structure. Like the mutation is happening. So that is the reason it is very hard to make the drug, because it is always changing its pattern. So for this reason, using the bioinformatics software tools, or still now people are developing some algorithm to compare different strains, like some strains already sequenced in Bangladesh recently, maybe till now maybe 20 to 30, and other country they have already sequenced many strains. So comparing those, like compares all these strains, we can find out why it was originated. Actually, it was originated from China, but we know, but actually it could be originated from another country. But no, actually it is originated from China based on this sequence. So from these evolutionary things, we understand that from where it is originated, how it is moving from one country to another country. Like in Bangladesh, from where it came, the virus came. It came from Europe, or it came from USA or China. We know that because based on the sequencing of those virus, we can understand from where it came to Bangladesh. So for this, we need a lot of computational things, computational power, like the cluster, which is we call the supercomputer things. So analyze those data. Also, some sophisticated tool using the cluster. But on the other hand, I told that the virus genome sequence is like 32 kilobase. But on the other hand, human genome, that is called DNA. We always heard the DNA sequencing it is using for different purposes. Nowadays, we can identify many things using the DNA sequence. So, but this DNA sequence is actually combined. When we do the sequence, it is combined of a lot of letters, like TCGA, these 4 letters. But from this 4 letter, we need to understand the patterns. But human, even there is no exact similarity between 2 men. Even both are healthy. But in the case of COVID infected people, if we compare with the healthy people, then we'll get some difference. So we need to identify those. But on the other hand, different coronavirus infected people will show different patterns. So it is very hard to compare and to find out, are they really based on DNA sequence, are they really infected in which level or infected by which strain? So why it is complex? Like if we consider any tissue, or even if we say simply from 1 drop of blood, and from 1 drop of blood, if we take 1 drop of blood and if we do the sequence using the next generation sequencing machine, we can get 30 GB data. From 1 drop of blood, it has 30 GB data, because in your DNA, everything is written what you are doing in every day. If you do something wrong, it is written in your DNA. If you smoke, it is written in your DNA. If you are giving some bribe to the people, it is written in your DNA. So there is no ways you can hide anything from your DNA. So from 1 drop of blood, it is making 30 GB data. And we know that from human genome, human DNA, that is the 3 billion base pairs, like the 3 billion base bits, like on bit, A, TCGA, like 3 billion. So it is not easy for a clinician or doctor or even anybody can understand from this sequence pattern. It is very hard. So from one sample 300 GB data, which we cannot analyze those data using our simple computer, personal computer. So for this, we did use some high configuration like cluster supercomputer things, and also some computer like computational power, like also algorithm to analyze this data. And this is doing by the people of bioinformatics research people are doing analyzing this data, and not only the analyzing this data, interpreting this data to the doctor, to the everybody. Okay, what is happening? What is showing this data? So there, why this is the reason the bioinformatics is very important field nowadays for health sector, also others as well as other sectors. And like before going to the next slide, I would like to explain a little bit about the central dogma. When COVID-19, like SARS-CoV-2, actually SARS-CoV-2 is a virus, and COVID-19 is actually the patient. So when SARS-CoV-2 interact with the human, then actually it is happening in protein level, not the DNA level. So what is happening? So DNA, what is DNA? We know the DNA. I explained a little bit about it. And after the transcription, it is happening RNA. Like if I say something, if I would like to do something, then it is like the DNA gives some message through the RNA. So it is like the RNA. And after the RNA, when it happened, then it is like protein. So it is that after the transcription and translation, it is becoming the protein. So in COVID infection, coronavirus infection, it is interacting with the human in protein level. So there are a lot of data nowadays producing for the drug identification, drug development, vaccine development, and that is trying to develop based on different kinds of data. Whole genome sequencing data, RNA sequencing data, chip sequencing data, and how it is producing. It is producing from the virus interaction with the human, infected people, how it is changing, how it is affecting. And these data we can analyze using different algorithm. We are developing different algorithm. We can analyze different ways, and we can like there are many things: copy number variation and mutation, gene expression, gene fusion, many kinds of data. So these data need to analyze for all these categories of prediction, identification, like drug prediction, or to use the existing drug for any new disease. And but another point is like, okay, so COVID-19 infected people, if we see different people are different, so it is not easy to understand these things. So that is the reason it is making delay to develop some drug or vaccine, because the virus is changing its structure. Also, when the human is infected, then it is making different type of problem for different people, not in similar way. So all these things we can analyze using the bioinformatics tools, and bioinformaticians can do the analyzing those data. Recently, so based on the actually prediction model, it is not based on the real model, the real data. So from one team from the USA, they found that, okay, from the clinical perspective, there are different diseases, patients with different diseases, they suffer more, their severity is more compared to the healthy people. So they found that actually the genetic patterns are different of those patients compared to the healthy people. That is the reason the severity is not similar for all people who are infected by COVID-19 infection. But it is not based on the real data. So they assumed based on their computational model and modeling of, but in the back end they use all the disease data, genetic data of different diseases. Based on this idea, we have started to do the research. Some of our research paper already published. Now we are writing almost writing and submitting almost 20 journal articles, and some are under review. And in our team, there are some people from the Green University, including Humayun Kabir Rana. He is also working with our team. Also, Professor Zaidul Islam, he has also a group of team members. He is also collaborating with our team and with me also to work on this coronavirus infection research work. So I'm presenting some of our work that we have submitted and some were published recently on this COVID-19 infection related work. So for example, in this work, we have tried to find out the genetic pattern of the infected people compared to the healthy people. So why it is important? If we understand that, and then we can identify the drug target. So drug target means, like, for example, some genetic marker, and using those genetic marker, then another group, they can develop some vaccine or drug. So this is the approach for developing the drug. So in this case, at first we have compared our new COVID-19 infection, like the virus sequence, with the other virus sequence. Like other virus means including SARS, MERS, which is similar to the, they are also coronavirus, but also the influenza, we have considered those to compare with the new coronavirus. And we found that actually it is similar to the old coronavirus, but there are some difference. But one new thing, but I'm explaining it a bit early, but one very interesting things we found that in new coronavirus, there are something which is little bit strange. Like we found there are something like HIV-1, strain of HIV-1 is included in the new coronavirus. Also the parasite, like the malaria, some part of that one is included there, which is little bit really strange. It is not the as usual things for the any virus. So, but it is different things. So in this case, we have considered the healthy people information, like the transcriptomic data, gene expression data, also the COVID-19 infected patient data. So those samples are actually the lung epithelial cells related, because when the virus infect to the people, then actually it attack to the lung mainly. It is respiratory disease. It makes respiratory problems. So then after finding those drug target, we found like 108 genes that are really significant. And based on that, actually people are thinking, not out from our study, maybe other people also found similar things. People are thinking to develop that drug. So now we found the 108 drug target, but we need a lot of validation things. We have done some bioinformatics approach. We have applied like the Fisher exact test, hypergeometric test to find out that are they randomly coming in our analysis, or they're really important for the COVID-19 infection. In this case, we need a lot of data. Maybe this sample, like here we have used like 6 samples, and for generating these 6 samples, we need like over $1 million. So this is on study. Actually, we have submitted this on, like it is under review in Science Journal. And second work, actually it is running by Humayun Kabir Rana of Green University. He is the leading author of this work. He is looking, okay, is smoking is a risk factor for COVID-19? So we have developed this project. Actually, still it is under the development. We will submit soon this paper. I hope we will submit in top priority journal. And in this case, we found that, okay, for smoking, actually, it is making some problem to the people, like it is affecting to the lung, and it is changing some genetic pattern of the lung, of the human lung. When the SARS infected, like if on smoke, who smoke, if he infected by the COVID-19 and another healthy people infected by the COVID-19, the genetic pattern of these two patients is different. And in this case, the problem, the severity problem of the smoker may suffer more than the healthy. Like healthy means like who is not actually smoker. So in this case, we try to find out, okay, if some smoker is infected by the COVID-19, maybe he or she needs different type of drugs compared to the usual people. So that is the reason we are trying to develop, we try to develop this model. In this model, similar, we have tried to find out what is the shared genes for the like synergistic genes is making, that is shared between the COVID-19 infection and also the smoking. And then we trying to find out the pathway and drug target and the validation things, function analysis using different sources of data. So another interesting thing that is like we have submitted to the World Psychiatric Journal. Its impact factor is over 40. It's like actually we found that psychiatric disorder patients, they suffer more if they infected by the COVID-19, like SARS-CoV-2. And especially we found that bipolar, schizophrenia, and post-traumatic stress disorder people, they suffer more psychiatric problem and they face severity for this infection. So in this study, we have tried to find out which genetic mutation or which genetic pattern is changing or making more influence for these infections. So I'm going quickly. So on paper is in our like also Human COVID Rana is also involved in this project. It is in the final revision. We have submitted the final revision. Hope it will be come out within 1 or 2 weeks. So in this case, we have, before coming to the real data of coronavirus infected, we like in a couple of months before, then we tried to find out, okay, the new virus is already in. So what is the like genetic pattern, which genetic pattern could be of the new virus? Before identifying the new virus data, then we did this work, and so it is almost going to come out the publication. So in this case, we have tried to compare all like different type of virus genetic pattern, and that is we made the phylogenetic tree, and we have also identified the gene expression, like identified the biomarker which are common, and similar way to find out the comorbidity, which disease could be like which disease patient could be affected more for the new coronavirus. And also we propose some chemical to develop the drug considering those chemical, like it is called drug compound or drug model, which could be useful for this novel coronavirus. So this is the like actually this is the this type of work we are doing for the COVID-19 project related to the bioinformatics work. We are also doing some different like machine learning and big data projects. And this big data and machine learning project actually considering the clinical data, symptom data, blood pathology, like blood test data. So on of our paper is under the review. In this case, like Akhtar ETL mainly. And in this case, actually it is first review. We found that there are some symptoms that could be important for the COVID-19 infection patient. So, and it could be useful for the early detection. Early detection, like if we feel some fever, it doesn't mean you are infected by the COVID-19. So, here fever, cough, fatigue, and many other things, in total maybe 8-9. Those symptoms are important for the COVID-19 infection. And considering those, actually we developed some algorithm, like machine learning algorithm, to identify which symptoms are really useful for early prediction. So similarly, we also consider the disease, like the patient who has the hypertension, diabetes, cardiovascular disease, how much they are affecting by the COVID-19. For example, they are infected by the COVID-19, but it could be make more severity who has this type of disease. So that is the reason the severity of the COVID-19 infection is different for different people. Some people are already dying because they have already existing some problem, like maybe the hypertension or diabetics or those type of problem. So they are not managing those hypertension or diabetics during this infection. That is making more severity and more problem for this infection. So in this case, our one paper already published in Expert Systems with Application. It is like Ahmad and like our team with Moni. So we published like we have started this work in early, like when we found that like China declared that there is COVID-19 infection is started, like at that time we have started this work. And for this we did not find huge data. So we have started to consider the Chinese raw data sets. And in our team, there are also some Chinese collaborators. They translated those in English language because we don't understand the Chinese language. And using those data, we have developed some machine learning model to find out the symptoms that could give us the early diagnosis. So without the real diagnosis using the PCR. So in this case, our model, like using this model, we predicted combined of some symptoms, not only single symptoms, or we have sorted out which symptoms gives the more priority, which like pneumonia, which is really important, could be maybe useful for the preliminary diagnosis, right, or preliminary prediction that he is suffering the COVID-19 infection. So yeah, so if you are interested, you can look at this paper in details. So and other papers like using the machine learning, we have submitted to some journals. So now, like before, we had only the very small data from the Chinese raw data sets. Now we have large data sets related to the symptoms, comorbidities, and severity death, like who went to the ICU, who already like recovered, and also the death patient information and different symptoms information, comorbidities information, because these are important. If we understand that these symptoms, he's suffering with pneumonia or ARDS or combined of those, then it is very like we are confirmed that he's suffering the COVID-19 infection. But if we see that some people are suffering like different type of symptoms, then maybe he's not suffering the COVID-19. This is our actually the second paper compared to the previous paper. So in this case, we use like over 500K patient information, but it was very hard to like collect those data and also to filter those data. It was very hard, and after filtering, we got only maybe a couple of thousand data, which are really useful for our model. And in this case, also we have considered the disease information, which diseases are really risky, like hypertension. We found that these are really risky for the hypertension people who has this problem. Also, second cases is diabetics or kidney disease, cardiovascular problem, COPD, asthma. Those people should take more care, and the government should give more priority to these people, and they should we need to give more priority to these people, because the severity of these people is more than compared to the general people who does not have this type of problem. So actually in this case, we look, okay, what is the like, there are symptom and hypertension. We found that the death people, the death patient, number of death patient, if we compare the pneumonia and hypertension, who has the hypertension and he's suffering the pneumonia is a symptom, and most of the cases we found that it was very hard to recover from this COVID-19 infection. So in this case, our machine learning gives the idea that combined of hypertension, combined of disease, like existing disease, and also the current symptoms, which are more important, which gives more severity. Why these are important? In case of distribution of the ICU, if in our country we have like 10,000 ICU bed, then if now the problem may arise like more than 500K patient, then who should give the more priority for the ICU bed? In this case, like pneumonia and hypertension, who has the hypertension problem and he's suffering, he has the symptom of pneumonia, he could maybe the more severity in this case. So I'm going to finish actually which type of research we are doing and which type of plan do we have to make some collaboration. Even we can do some collaboration with the Green University Bangladesh. So we are doing the early detection So early warning. So like similar to the smoke signals. In case of smoke signals, so we know that if the smoke signal we get in early signal before fire, like a fire alarm, so we can make some type of device. If you have some idea, we can develop some device which can be useful to give the early warning us. And also for diagnosis, we are using some CT scan image. Also, there's some clinical information like pathology test. Using the pathology test, we can predict the patient, and we can diagnose the patient without using the PCR testing. Also, the case of prediction case, like prevention, we can use some prevention approach, like calculating the probability of infection. Uh, Also, the information we are using, like we are developing one model, like using the information from the Twitter, so the news and Twitter, so we can understand the mentality of the people, mentality of the patient. Also, we can predict the mental situation of the or psychiatric situation of the infected people. So also some people are doing the like robot system to make the distance from the patient. Also some automation like chatbot, so to get the support from the people, or like the from the doctor, or some automated doctor like the AI doctor, to get some support related to the related to the infection, or related to the drug, or related to the any support from this AI system. And also we can monitoring, we are not doing this work, this type of work, but maybe some people are doing the monitoring system like using GPS, satellite system, and media data. So thank you very much.